My name is Natalie Gilbert. I am a center here at Pingry, and the title of the paper that I study is called Activated CLPP Kills Persisters and Eradicates a Chronic Biofilm Infection, or in layman's terms, treating chronic infections. I'll actually go on this side. Okay, so before I begin, I just wanted to acknowledge a few people. I wouldn't be here without any of the people on the board here today. First and foremost, Mr. D, for fueling my love for the sciences. I actually shied away from biology after taking his class. But once I came to him junior year saying that I wanted to try pursuing research, he took me in with open arms, and I've loved it ever since. Mark, the two Jacksons, and Claire for making the journal club process easy for me. This presentation was actually my first journal club presentation, and they made the process a lot easier than I thought it was going to be. So thank you to them. And obviously all of you, thank you for coming. It's truly an honor to be up here today. Yes. So why did I choose this topic? So my mom was diagnosed with cancer a couple of years ago, and ever since my cliche dream has been to find a cure for some huge disease that's going to change the lives of so many people. So I told Mr. D my dream, and he told me to start that dream by applying for an internship at a university over the summer. And so luckily I was able to land an internship at Princeton University with Dr. Mark Brynildson at the Department of Chemical and Biological Engineering. And he studies bacterial infections. So ever since, I've been absolutely in love with the topic. It's also my AP Bio project, and while I was researching, I actually came in contact with Dr. Kim Lewis, who's actually one of the leading professors in this topic. So, and when Sydney actually gave me the paper, I discovered that he wrote it. So it's particularly an honor to be up here today because this data is very groundbreaking information. It was recently published a couple of months ago, and it's really exciting, so I'm excited to share it with you. Okay, so to fully understand the mechanisms behind chronic infections and to really understand what they're trying to argue in the paper, we need to understand what a persister is. So a persister is basically a small subpopulation of bacterial cells that are tolerant to antibiotics. They're also characterized by something called a biphasic killing curve, which just means that they die much slower than normal active bacteria. So as you can see here in this graph, the bulk of the bacteria are dying immediately in response to antibiotics. But there's a very, very tiny percentage of bacteria that for some reason aren't dying immediately. And we don't know what characterizes this. We don't know what characterizes a persister. We don't know if it's genetically different to normal bacteria that are dying immediately. But we do know that they have the ability to enter a state of dormancy. I'm going to explain that in the next slide. So imagine that this plate of bacteria is a bacterial infection in your body. So for example, let's take cystic fibrosis. So your doctor is going to prescribe you an antibiotic to treat it. And that antibiotic is going to kill the vast majority of the bacterial cells because that's what it was made to do. But for some reason, a tiny percentage of that bacteria isn't dying immediately. It's tolerant to the bacteria. So it's entering this dormant state, which just means that it stops replicating. It enters this sleep cycle. And for some reason, it has the ability to sense when the antibiotic has left the body. And once it senses that, it then reawakens and resurges its population back to normal as if nothing happened in the first place. So this whole cycle is really what underlies a chronic infection. So if we can understand the mechanisms behind a persister, and if we can understand why they become dormant, we'd be one step ahead of figuring out how we can fully eradicate every bacterial cell that's involved in a severe bacterial infection. Okay, so now let's try to understand what a biofilm is. So all you really need to know about biofilms are that they're just a protective layer for persisters from the immune system. And they typically form on surfaces in the body like prostheses and heart valves. And you can't really see in this picture, but they form a slimy extracellular matrix as its form of defense from UV radiation or antibiotics. Okay, so the bacteria of interest in this paper is Staphylococcus aureus, otherwise known as Staph aureus. Staph aureus infections are not very prevalent in the United States. There are about 100,000 cases every year, and approximately 19,000 of those result in mortality. So obviously Staph aureus infections aren't as quote-unquote major as diseases like cancer, but obviously it's taking away the lives of so many people, so it's still an issue that needs to be addressed. The severity of Staph aureus infections vary. Some can be minor, like just having pimples or having boils, and some can cause life-threatening illnesses like meningitis, osteomyelitis, which is a bone infection, and endocarditis, which is a heart valve infection. Okay, so now let's try to delve into what they're arguing in this paper and what activated CLPP means. So this whole picture is basically their proposed solution. The top two circles represent the active bacterial cells that die immediately, and the bottom two represent the dormant persisters. So in the top left, I kind of mentioned it before, your doctor will prescribe you the conventional antibiotic, and that antibiotic is going to bind to a specific target in the cell, and it's going to corrupt that product. And then because that product is corrupted, the cell will self-digest and die. But the problem is with dormant persisters is that this target is for some reason inactive. And once the antibiotic tries to bind to that inactive target, well, it's inactive, so it can't create a product, it can't corrupt a product. So consequently, there's no effect on that cell. But what they have proposed is that there's a new antibiotic that they've just developed called ADEP4. And it has the ability to bind and dysregulate a regulatory protein called CLPP. A regulatory protein is basically a protein that regulates the activation and deactivation of many proteins within a cell that's important for its own function and survival. So once ADEP4 binds to CLPP, it dysregulates it, and immediately the cell undergoes a process called proteolysis. And proteolysis is just the breakdown of proteins. So suddenly, because there's no more CLPP, all of these proteins are going crazy, and because they have no regulatory protein to work under. And so the cell, therefore, self-digests and dies. And what they think is that the same exact thing is going to happen with dormant persisters. Does anyone have any questions so far? Okay. So they started to perform a few tests. So the first experiment they tried to do was to see what would happen if we exposed the ADEP4 to a plate of bacteria. And their results weren't expected. By day 2, the bacteria started to die, but for some reason, the bacterial population started to resurge by day 2. So then the next experiment that they tried to perform was to see what would happen if we exposed different types of conventional antibiotics to the plate of bacteria. And as you can see, there was virtually no effect in the death of bacteria. So the next test that they tried to perform was to see, okay, let's see what would happen if we combine two antibiotics. And what I mean by combining is to add one antibiotic for the first 6 hours, and then add a different antibiotic for the remainder of the experiment. So what they saw was that the combination of ciprofloxacin and ADEP4 led to the complete eradication of the bacteria. So the next test that they tried to perform was to see if there was any other combination of antibiotics that would prove to be more effective than the ADEP4 and the ciprofloxacin. And what they discovered was that the combination of ADEP4 and rifampicin, as you can see with this steep slope, that combination seemed to be the most effective combination of antibiotics. And this bar graph over here is just a representation of the other tests. The star on at the very far right just symbolizes limit of detection, which just means that they couldn't detect any more bacteria on the plate, which is really groundbreaking stuff. So after they performed all these tests, they were like, wait, we've been performing all of these tests on MHB broth, And MHB broth does not mimic that of a human body. So what they then decided to do was perform the same exact tests on an animal model, and in this case they used mice. So as you can see in the far right, there was virtually no change. As you can see, by day 3 they reached the limit of detection, which means that they couldn't detect any bacteria. So that's really amazing stuff. So they then tried to see what would happen if they basically tried to see if all the bacteria would die in different strains of the Staph aureus, because different Staph aureus infections aren't from the same Staph aureus strain, they're from different strains. So basically they just proved that with these 4 strains of Staph aureus, all of the bacteria died, which is amazing, which shows that this combination will be able to treat many different types of Staph aureus infections. The USA 300 strain is affiliated with soft tissue and skin Staph aureus infections, and I know that the UAMS1 strain actually represents osteomyelitis, which is a severe bone infection. Okay, so this basically just shows what the result would be if this combination of antibiotics actually worked on humans. Eventually, after applying those 2 antibiotics, it would hopefully lead to a happy and cured person. So why should you care about this? Our antibiotic arsenal is becoming obsolete, so it's extremely important that we understand the mechanisms behind bacterial persistence and biofilm production, so that we can create novel treatments out of what we already have. And these set of experiments will undoubtedly serve as the future model for the killing of other proteases that are implicated in life-threatening cancers and bacterial infections. So, yeah, that's my presentation. Does anyone have any questions? Yes?
Has the product used?
No, this data is very recent, so they haven't actually. But that's definitely their next step.
Study the effect of that combination of antibiotics on beneficial bacteria? Is it destroying non-targeted bacteria?
I'm sure it is. So that's definitely something that they need to research. Yeah.
How many people involved in the study? Yourself,
I didn't actually do this study, but I know that about 5 people were focusing on this study. Yes. Okay, thank you very much. Thank you.
Okay. That is good guys. That's like
No less than
you know.
So the underlying these guys, like this one particularly, just the presentation that comes later. Those are the 2 skills we want to teach basically this. So ideally you take kind of all parts of this and each part is reinforced. Each part reinforces one of those skills. And then generate research the best scientists in the world can explain it.
No, my friend tries on which is communicate whatever it is you invented or otherwise this stuff.
Exactly.
Yeah.
That's what I tell them. Like, it doesn't matter how good with your science you are.
Yeah.
Whatever you invented, if you can't replicate that, you know that.
All right, no longer go.
Hello.
Yeah. Good. How are you?
Alright.
Am I going to send you? No, I'm not going to send you.
You were considering.
Oh yeah. I was going to go to LA. I applied for a job in San Diego. So it's called Tech High. But I think I'm excited.
Okay, you get that anyway? Because I definitely
I
have
got
that
this.
on my side. Well, maybe you want to like try different cases and then try it that day.
But you're thinking teaching again?
Maybe try that.
Is that a done deal?
That'll be the time I drop it.
Pretty much.
Today I switched it. Today you switched it. Oh, you know what's actually really good?
It's great.
I just like the kind of solid rubber ones. If you drop it,
Well, you know,
it shocks a lot.
working in the cubicle,
It's not trying to say if you use soft rubber ones, it's actually big.
it's great for a few years, but I mean, I got the 10 years out of professor.
The soft ones absorb it a lot, so if you want to try it, suggest it. Yeah. Because I tried one.
It's not probably not the most,
The hard ones.
it's not MIT or Harvard, but it's okay.
But you're really racist. You can take whatever.
And try to
It
get
will
one
help me.
Yeah. Yeah. This one has battery. And I used to have, the best one I had was like, it's a soft
Well, not so
base,
much,
but they have
but
a
yeah.
hard case. It's like
You know, if you ever want to do collaboration here, always.
Yeah, maybe. Yes.
I think less than this.
We have up here, see the,
If you want to go to one that's like less.
so we're trying to get that program on
You know, I hate to move away because something that's not going on, but
I drop it all the time.
no, it's
I think first thing is test.
roads are the
I have one exactly like that. I got it like
It's a
Hong
big
Kong.
tourist destination, it's like very nice destination.
It works well.
But,
I didn't even realize it.
you know, I couldn't, the only, I mean, I applied for positions here.
It's external.
Or you can change it, you can change it, charge together all
What they need, so,
Yeah. I would love that.
very respectful.
That is
It's really trying.
as long as that's turned
Yeah,
on.
it's very close to that.
But there's something wrong. What?
Was there a lecture here?
There was.
So, okay, so we'll go to the.
Yeah, and also make sure like,
That's all fine, you know, it's not really good to go as
it's also heavy. It's so heavy. Well, that's not like that. Yeah, it's like,
Then it's down and there you go.
mine's even bigger than
I mean,
I think that's brand. So if I get one brand, I want
If you really get used to
it's
it.
gonna be a rough start.
Yeah.
Yeah.
My mom just wanted this for
I don't think so.
And it's like
I've done it before.
Yeah,
For
mine
the
is so
five,
clunky.
for the five, it's
Mine is so clunky.
So you're next
It looks like a 5 because it's like longer, but it's like heavy.
Well, it's a bit of a disconnect. My old
I got
habits.
this like life proof one. I don't know if it's life proof one, but I got it off of
But
eBay.
I would have to do it again.
I'm just trying it out. No, I haven't tried
You know,
it.
it was
It's
kind
like $100, so I
These
don't
people
know.
need reviews.
Yeah, I'm gonna try that.
We just all need to group up there.
No, I never had a group. I mean, I was working in
Still don't quite understand.
And I'm starting to get some stuff in sketches. It's like 3 weeks ago.
I was an employee and
No.
I was the manager.
Oh no. But I thought it was good. Yeah. So when I saw it, you know,
It was who
kind of
scored.
imagined that. Yeah.
We all just,
Was it or something?
Yeah.
we all like, we were all very good.
No, it wasn't that.
No, after a couple of years of this, you can't spend your whole life.
Resell.
Yeah. And even though it was a new
Yeah.
And besides my interest, I couldn't change, you know, I want to do some other things.
Amazon's the best. If you want to go to Amazon.
You know, my father is like,
There's always like things for like buyers, sellers, and like
I still don't understand what you're doing.
Maybe it's like contact with someone.
yeah, because like
I have to go.
Okay.
Yeah, you're presenting in 10 minutes in that room.
Right.
you're on your own there.
Thank you.
I feel like I paid
Can you go first?
that's not that
I'm gonna bring you the-
Hey!
PayPal's good so it wants to verify information, but if I don't get your money back, you should still call me. Call the seller first, and then if they like, they don't answer, so then call me.
looking for
I know someone, I don't know if it was a joke
all right,
or something, but I know someone who like sold a weed box for 3 years ago, and it was like, it's a weed box on it, and everyone was selling it and bidding on it, because they thought the weed was inside it. It was just the weed box.
But it said weed box though.
Yeah, but instead of weed box, so like they couldn't use
Yeah,
it.
so they can't hold them to it, right?
Yeah.
It's the same thing.
Worse than weed box. You gotta watch out. You gotta watch out.
You know, you really do. Tickets, you really have to be careful. I know some
StubHub is good about that. StubHub is really good about that. That's what I.
They'll guarantee you, right?
And if it doesn't work, they'll either exchange it or reimburse you. Or they also charge a commission fee for the other person, not for you. And like a service, doesn't have $50 or whatever, but like it's basically warranties.
They got games where you find a basketball. And just recently, bought the tickets online, and now you have sport tickets. When they're all excited, and they're particularly down there. So you really have to.
Oh my gosh. Were they able to like,
I don't think so.
oh my gosh.
You really
You lose a
have
lot
to.
of money anyway.
And you've gone, like they came from Virginia or something to come into New York.
And they couldn't get in.
Yeah.
Wait, so did they not know where to like?
Yes, they did not know because they look like regular tickets and everything.
They couldn't scan it, right?
And then, yep, and then when they got their answer, they were told these are.
Hi everyone, our final career speakers are starting in 10 minutes in room 302 and 306. Hope to see you there.
Yeah, but we went to like Ticketmaster, obviously, Live Nation, and like StubHub is good, obviously, and games too. And I heard tickets now. I mean, I've had 2 like tickets to clubs that I can't go to, so I'm trying to sell them.
So are you trying to sell?
Yeah, I'm trying to sell, but like for real. I promise.
What is it?
Avicii, have you heard? They like sing "Wake Me Up" and "Hey Brother." I'm probably going away with the summer. It's on the 28th of June. So yeah. Thanks, Natalie. Hello.
Hello. Is there something going to be happening in here?
There's going to be a speaker on here.
Oh, okay.
We're going to be here tomorrow to the concert.
Good press.
7 days a week.
But you're almost done with that.
I know.
All she didn't say, she said to like me the other
Oh, there is something like that. I don't understand. Ew, how do you think they are?
You're not going.
Yeah, it is mostly parents. There's actually a lacrosse game,
Oh, that would be.
so most of the kids kind of ditched.
You need to go make sure Nana and there's in case.
No, I'm gonna like.
Yeah, because I'm like, do you have someone like just waiting there?
I know, but it's. I think because we.
No, you need to listen. It's your thing.
Come on in.
Yes, we do. First, we have cleanup.
Right.
Then, yeah.
Oh, you mean today?
Oh, yeah, today. But then we have, if you want to email me,
I mean, I can hang out.
yeah, I don't know, I'll have to ask the
If
they want it. Yeah, exactly.
No, I'm sure.
I mean, if it's parents, it's different, but if it's
Exactly.
they want.
Yeah.
And it doesn't have to be this event, it could be maybe a journey. It's not necessarily, you could have to be. I'm interested.
Yeah.
I want to lead to it, and I want to help with that. And you don't want to. Yeah.
Yeah. We would love you to come.
I love that. Or
Yeah.
I don't know.
Yeah, I have.
Your father's mildly looking for you. I think you're just curious where you are. Yeah, and then like the act, like closing remarks are at 3 o'clock. Yeah.
You know, Luke told some of the other people they had to have music in their rooms.
What?
Just tell
You
them.
didn't bring a guitar today.
They were specifically instructed they have to have music in their rooms.
I found that a little distracting.
He's a musician in his own life.
Yeah, it's a crack. I was asking, it seems like there's just mostly parents. She's like, yeah, I think there's a lacrosse game at the same time. I pulled some of these students that are interested.